Okay, so continuing with the second part of the neural coordination chapter, we will go into the details of two specific sense organs in this chapter. So, as I mentioned, that we have five different sense organs, namely the eyes, the ears, the nose, the tongue, and the skin, out of which we will get into the details of the eyes and the ears in this particular section of the video. Okay, so this is the structure of a eye. This is the transverse section of a eye that you see over here. And first, let's begin with what are the different layers in the eye. So the outermost layer is called the sclera, the layer next to it is called the choroid, and the layer next to the choroid is called the retina. So you can see it marked over here. This is the sclera, then the choroid, and then the retina. Okay, so what is the sclera? The sclera is a very dense connective tissue layer, whereas the choroid consists of blood vessels. Okay, so the middle layer in the eye, the choroid layer, this has blood vessels that supply blood and oxygen to the eye. And then the innermost layer, that is the retinal layer, this composed of the ganglion cells, the bipolar cells, and photoreceptor cells. Okay, so the retina is basically what detects the light that comes into the eye and helps us in visualizing our environment. Okay, so these layers are named as the sclera, the choroid, and the retina at the rear part of our eye, whereas when we move into the front part of our eye, so this is the rear part of our eye and this is the front part of our eye, then the sclera in front becomes the cornea, and then the choroid, see the choroid layer over here in front becomes the ciliary body. So this particular piece of structure that you see over here, this and this, so the choroid layer comes in front from the ciliary body, and in front of the ciliary body, further in front, is called the iris, which is the pigmented and opaque part of the eye. So if this is the ciliary body, then this is the iris of the eye, and this iris controls the amount of light that enters the eye. So the iris is basically the pigmented part of the eye. If you look at somebody's eyes and you see that some eyes are, let's say, brown in color, some may be green in color, some may be blue in color. So the white, the colored part inside the white part of the eye that you see, that is basically the iris. And this is the diameter of the iris. So this particular opening is the diameter of the iris, and this controls the amount of light that is entering into your eyes, right? So let's say suddenly you focus a torch on the eye, and then too much light goes into the lens, and this can damage the retinal layer behind. So immediately this iris comes forward and this opening is narrowed so that less light can enter the eye. So you can do this experiment at home yourself. So you can take a torch and you can take a human subject, take your brother or sister, or maybe even your mother or father, and tell them to open their eyes and then focus the torch on their eyes and suddenly switch it on, and you can immediately see that the diameter of the iris narrows or shortens down, so that this opening that allowed light into the eye is narrowed and less light enters the eye. Okay, so what have we come to know? The middle layer of the eye, that is the choroid layer at the front part of the eye forms the ciliary body, and then the ciliary body moves in front to form the iris, and this iris controls the diameter of the pupil, this opening that is also known as a pupil, and it controls the amount of light that enters the eye. Okay, then what is the function of the ciliary body? this ciliary body basically maintains the extent of convexness of this lens. Okay, so this lens that you see in the eye, this is a convex lens, right? And I think people who are from physics, you know that a convex lens, let's say, looks like this. So a convex lens has a particular focal point, right? So if this is a convex lens, then if light enters it, then it focuses at a particular point. Okay, so this particular point of a convex lens is its focal point. Now in our eye, this focal point is basically the point of the fovea. This is the fovea, also known as fovea centralis, and this is a depression in the retinal layer of the eye where we have the maximum visual acuity. So a term is known as visual acuity. This is a point where we can where we can see objects with the maximum clarity. So when you are seeing something, let's say you are reading something from paper and you suddenly take your eyes up and let's say you focus your eyes on something further away, let's say a calendar that is far away from you. So immediately you can notice that it takes hardly a fraction of a second, but you immediately can focus on the writing in the calendar from, let's say, reading the notes in your copy. So what is the lens doing, or what is the ciliary body doing at that point? It is basically changing the diameter of the lens or the extent of convexness of this lens. Okay, so normal glass lenses, they have a fixed shape, right? So a glass lens is like this, but this lens inside our eye, which is made of particular tissue, can be shrunk to form a lens like this, or it can be expanded to form a lens like this. And this movement of the lens is controlled by this ciliary body. And why is the lens shape changed like this? This is just to change the focal length of the lens. So because if we have to read something, we need to see it with the maximum clarity. So whenever we need to see something with maximum clarity, we need to have the light coming from that source to be focused on this particular spot in the retina that is known as the fovea centralis. And whatever light that falls in this part, we can read that with maximum clarity. So when we are reading something that is close by, then the lens has to take a particular shape so that the light from all the closed areas focuses on this particular point. Whereas whenever you read something that is far away, so immediately the focal length of this lens is changed by changing its shape, and now you can focus on whatever is far away, and all the light from that far away object again focuses on this particular point on the retina that is known as the fovea centralis. So that is there. And the retinal layer again consists of certain photoreceptor cells. So as I was telling, the fovea centralis is the point where we have the maximum visual clarity, also known as visual acuity, which is because and the entire retinal layer is composed of rod cells and cone cells. These are basically named rod and cones because the rod cells are shaped like, say, like these, and the cone shells are actually shaped like cones like this. So that's why the name of rods and cones comes. And these rod cells are involved in twilight vision or vision in low light. So let's say when at night, when the lights in the room are turned off, then also you can adjust your eyesight and then see objects around you with some amount of clarity, right? It does not appear completely dark. You can still make out the objects. So that is brought about by the rod cells. And during daylight, you use the cone cells, and in the cone cells produces photopic vision, also known as color vision. So at night, when you see in low amounts of light, then you do not see color as such. You do not see the shades of red, green, or blue. All you see are shades of black and white. That vision that you have at night is due to rod cells, and they have a pigment protein that is known as the rhodopsin, also known as visual purple. And in the cone cells, there are three different kinds of cells, three different kinds of photoreceptor cells. One is for red light, one is for green light, and one is for blue light. And when all these three different kinds of cells are stimulated all together, then you have the sense of white light. So white light, as you must have read from physics, is a combination of all different colored light. So the lights, let's say in the rainbow, if they are combined together, then they form white light. So similarly, light is detected in a similar manner inside the eye by the cone cells. So the cone cells, which comprises of the cone cells have different pigments for red light and green light and blue light. And when all the different pigments are stimulated all together, then you have the sense of white light. Now, when you read something, or when you focus on a particular object, then the light must be focused on this particular area of the retina, that is known as the fovea centralis. So this region has the maximum concentration of the cone cells, and the surrounding regions, in these peripheral regions of the retina, the rod cells are uniformly distributed. And these rod cells are much more sensitive to light than cone cells. So very minute amounts of light can also stimulate the rod cells, the photoreceptor cells, and thereby it can help it to produce some sort of an image of the environment even under very low light conditions. This is due to the presence of the rod cells in the peripheral part of the retina, which are extremely sensitive to light. So even very small amounts of light can trigger these rod cells and help us to have some sort of vision of our surroundings. Okay, then another thing that you see in the eye, this is known as a blind spot. The blind spot is basically a gap in the retina. So this is the point where the optic nerve leaves the eye. So this is basically the eyeball, and then whatever light came and hit the retinal layer through this apparatus of lens and everything produces nervous stimulation, and this nervous stimulation travels down this optic nerve to the brain, which produces a sense of vision. So this particular point in the eye where the optic nerve leaves the eye, there are no rod cells and no cone cells. So if any light that comes in this particular region is not at all detected by the eye, therefore it is known as a blind spot. So in the blind spot there is no rod cells and no cone cells, and hence there is no sense of vision if any object is focused at the blind spot. Okay, so that is there. And then one more thing that I should also mention at this stage, that this lens and the ciliary muscles divide the front part of the eye from the rear part of the eye. So the front part of the eye is also filled with a fluid matrix that is known as the aqueous, it's mentioned chamber over here, it's also known as aqueous humor, and the rear part of the eyeball is filled with another matrix like substance. This is known as the vitreous humor. Okay, so now you have an idea about the structure of the eye and a basic idea how vision is produced. So the light comes in through the pupil, the diameter of the pupil, which is controlled by the iris. So the iris has certain muscles which can regulate the diameter of the pupil, and in turn determine how much light enters the lens. The lens focuses maximum part of the light onto the fovea centralis. Light is also focused in other parts of the eye, but whatever light focuses on the fovea centralis, those regions we have the maximum visual clarity. So I'll give an example of this. Like, let's say you are right now maybe seeing the video on your mobile. So you are focused on your mobile screen, but you do have a sense of peripheral vision, right? Like, so let's say if your mother walks in around you right now, or if anything happens in your surroundings, you do not see it very clearly, but you do have an idea of whatever is happening in the surrounding. So that is known as the peripheral vision of yours. And so that is because whatever the light hits the remaining part of the retina other than that hits the fovea centralis, that creates the sense of peripheral vision. So you do not see those things very clearly, but you do have an idea that what is happening around you other than the particular point where you are focused very sharply. So you are focused very sharply on your mobile screen, so whatever light from your mobile screen is going to the eye is focusing at the point of fovea centralis, but the area around you is also being visualized by the eye continuously, but you do not see it very clearly because those lights are being detected by the peripheral part of the retina. Okay, so this is regarding the structure of the eye and how the light is detected by this by the eye. Now let us get into a little more detail regarding how the vision is created in the eye. So vision is created due to the splitting of the photopigment, that is known as rhodopsin. So in the rod cells, the pigment is known as rhodopsin. In the cone cells, the pigment is known as photopsin. So the word photopsin is not mentioned in your book. The pigments are almost similar in both the rod cells and the cone cells, but they obviously have other differences because the cone cells help in production of colored light vision, whereas the rod cells only produce a sense of black and white vision. So when light hits this particular pigment in the rod cells and the cone cells, the photoreceptor cells of our eye, the pigment is split into a protein that is known as opsin and into another compound that is known as retinal, which is an aldehyde of vitamin A. Now, the dissociation of rhodopsin into opsin and retinal, and also similarly the structure of opsin is also changed by this reaction, and all this bring about a change in membrane permeability. So this change in membrane permeability results in a potential difference in the photoreceptor cells. So initially the membranes remain extremely permeable to sodium ions, and whenever the light induced dissociation of the rhodopsin pigment into retinal and opsin occurs, the membrane permeability changes and the membranes do not remain that much permeable to sodium ions, and therefore a potential difference is created in the photoreceptor cells, which generates action potentials, and these action potentials are transmitted to the visual cortex in the occipital lobe by the optic nerve. Okay, so and also remember, as I mentioned in the previous part of the last video on neural system, that there are bipolar neurons are present in the retina of the eye, and that vision is detected by the occipital lobe of the cerebrum. So cerebrum has two cerebral hemispheres, the front and each hemisphere is divided into four lobes: the frontal, the parietal, the occipital and the temporal. And the occipital lobe was that one which was involved in the visualizing process of our eyes, and so all the sensory signal that is generated because of the splitting of the pigments and the change in membrane permeability and the potential difference, that is carried by the bipolar neurons to the occipital lobe of the brain, and it analyzes the signal, and thereby you get a sense of vision, or you get a sense of your surroundings. Okay, so all this is regarding the eye and how you have vision of a particular object through the eye. So the next sense organ that we will concentrate upon will be the ear right now. Okay, so this is a human ear that you see over here. Okay, so human ear has three different parts. Let's say the outer ear, the middle ear, and the inner ear, right? So the outer ear consists of this ear pinna. This is ear pinna, basically the part of the ear that you see outside. So this is the ear pinna, and then the ear canal. So this ear canal is through which the sound waves enter your ear. So all the sound waves, so let's say right now the sound that you're hearing, so that sound that is coming out from your mobile speakers is hitting, the sound vibrations are hitting the ear pinna, and then this ear pinna basically directs the sound waves into the ear canal. Okay, so this ear pinna and the ear canal constitutes the outer ear, and they help in directing the sound waves towards the inner ear. Okay, and in the inner ear there is this eardrum. This is also known as the tympanic membrane, and the sound vibrations that are directed by the ear pinna into the ear canal to the tympanic membrane or the eardrum causes vibrations in the tympanic membrane, and these vibrations are transmitted into the middle ear. So the middle ear basically consists of three different bones. So these are bones known as the malleus, the incus, and the stapes. So the stapes is also the smallest bone in our body, and one more thing, this eardrum, also known as tympanic membrane, is also part of the outer ear, and the inner ear consists of these three bones, the malleus, incus, and stapes, and this lie in the cavity. This is known as the tympanic cavity, and then they. So sorry if I mentioned it wrongly, I'm not sure. The malleus, incus, and stapes form the part of the middle ear. Okay, the three bones, malleus, incus, and stapes. So you can see this is malleus, this is incus, this is stapes. So these are basically three bones. So this is first one is malleus, then there is incus, and then there is stapes. And these three lie in this particular cavity. This cavity is known as the tympanic cavity, and these are parts of the middle ear. And after this middle ear lies the inner ear. This inner ear is also known as a labyrinth. Okay, so these structures that you see over here, these are parts of the inner ear, and they have the bony labyrinth, that is the outer part of this structure. It's made of a bony material that is called the bony labyrinth, and the inner part of this structure, this let's say this hollow snail shell type of structure. So if have you seen a snail has a shell, right? This inner ear also looks like that, and this outer part is bony in nature, therefore it's a bony labyrinth, and this shell is divided inside by membranes, and they are called the membranous labyrinth. Okay, one point I should mention over here, that this middle ear is also connected to the pharynx, and this connection to the pharynx is through this tube. This is known as the eustachian tube, and this basically helps us to maintain the ear pressure, right? So you remember the pharynx, that pharynx is part inside our mouth, the nasopharynx and the oropharynx. The oropharynx through which you swallow food, and the nasopharynx through which the air that you're breathing from the nose passes by. So the ear is also connected. Our ear is also connected to that pharynx, right? Through this eustachian tube and through this middle ear. So remember, when have you felt? Some of you might have felt this. I'm not sure. When there is a change in pressure. If somebody has traveled in a flight in a plane before, when the plane reaches a particular altitude, or let's say even when you go to hilly regions, sometimes you feel that your ear drum is blocked. You feel a sort of a weird feeling in your internal ear, and that is because of the changes in air pressure. Because let's say when you reach a higher altitude, then there is less air pressure over there. So the pressure on this side of the eardrum and the pressure on this side of the eardrum should be same. When there is a difference in this pressure on both sides of the eardrum, then you feel that weird sensation. And then they say that if you open your mouth and try to take in some air, so then that sensation is relieved. That is why, that is because the air from your mouth and then the pharynx travels through this eustachian tube and reaches this part, and then the air pressure on both sides of this tympanic membrane are cancelled out, and then that awkward feeling that you feel inside your ear due to changes in elevation is ameliorated, and you do not feel that sense of discomfort anymore, right? So That is a function of this particular eustachian tube that you see that connects to the pharynx and then to the mouth. Okay, then let's come to this inner ear. This inner ear is one of the most complicated and most beautiful organs probably. So as I mentioned that the sound waves that are coming in from outside first creates vibrations in the tympanic membrane. The vibrations are transmitted by these three set of bones, the malleus, the incus and the stapes, to the inner ear, and then this inner ear is involved in detection of the sound. So how does it do that? So let's concentrate on this particular part of the ear over here. So over here, the stapes, the smallest bone in our body that is also involved in transmission of the sound waves, is connected to a part that is known as the oval window, right? And there is another opening over here in the inner ear that is known as the round window. Okay, now if you have a cross section and if you look inside what this particular organ, this is also known as a cochlea, so which looks like a snail's shell, right? So if you look inside this cochlea, there are divisions in this through two membranes. What are those two membranes? They are known as a Reissner's membrane, and then there is known as a Basilar's membrane, right? So there are two membranes that pass through this entire part of the internal ear and divide the space that is inside over here into three compartments. What are the three compartments? They are known as a scala vestibuli, then there is known as a scala media, and they are known as a scala tympani. Okay, so this part is known as a scala vestibuli, then this part, the yellow part, the blue part, the first blue part that you see going, right? The starting from here, let's say starting from here, this is known as the scala vestibuli, right? So this blue parts you can see marked as scala vestibuli, and this is the scala vestibuli. So this cross section is again zoomed over here, and this middle part in the yellow region, that is known as the scala media, and then the next part is known as the scala tympani. Okay, and this division of the inner ear into these three compartments is brought about by the Reissner's membrane. So the top membrane is known as the Reissner's membrane, and the bottom membrane is known as the Basilar's membrane, right? And then another thing is that this scala media is filled with endolymph. So the endolymph that you can see colored in yellow throughout, and then this scala vestibuli and the scala tympani, they are filled with this blue colored fluid that is known as the perilymph. So the fluids are not actually colored, it's just that so it is easier to understand, it is marked with the blue, yellow and the blue color again. So these blue colored regions are that of the endolymph, and the yellow colored regions is the perilymph. Okay, so just summarizing once again, the Reissner's membrane and the Basilar's membrane divide the entire space in the internal ear into three different compartments. The three compartments are known as the scala vestibuli, the scala media and the scala tympani, of which the scala vestibuli is filled with perilymph, the scala media is filled with endolymph, and the scala tympani is filled with perilymph once again. Okay, now so how does all these organs help in the transmission of sound waves and detection of sound through the brain? So as I mentioned, the steps that is connected to the oval window transmits the sound into the internal ear, and this causes the vibrations in the perilymph of the scala vestibuli. Okay, so as you can see, this stapes is connected to the oval window over here, and this causes vibrations in the perilymph of the scala vestibuli. So the vibrations are then transmitted from this particular perilymph to the Reissner's membrane and then onto the endolymph. So this perilymph causes vibration of this Reissner's membrane, and this Reissner's membrane causes vibration of this endolymph. So the vibrations of the endolymph causes movement of the hair cells that are present in the organ of Corti. So this particular structure that you see over here, this is known as the organ of Corti. This organ of Corti consists of these hair cells over here, and then this membrane, this is known as the tectorial membrane. Okay, so the sound vibrations were passed onto the endolymph, then onto the perilymph. This perilymph caused vibrations of these hair cells in the organ of Corti. The hair cells then touch this tectorial membrane, and this generates certain nerve impulses again, and those nerve impulses are transmitted through these cochlear nerve fibers into your brain, and then your brain detects the sensation of sound, right? And then what is the function of this last endolymph? That is the scala tympani, and that is part of scala tympani. So when we have very loud sound, then the vibrations are transmitted from the endolymph also onto the perilymph, and these vibrations are then transmitted back into the round windows. Okay, so vibrations are transmitted from this endolymph layer, this perilymph layer into this endolymph layer, and if the endolymph layer basically detected the sound, and if there are very loud sound, the extra vibrations are transmitted into this outer perilymph layer, and this perilymph sends the vibrations back through the round window. So these sound waves are backed through the round window to the middle ear, and then this middle ear transmits the sound towards the pharynx. Basically, this is causing the dissipation of the sound waves, because if there is a too much loud sound, then it might cause damage of your membrane and damage to your hearing apparatus. So this extra vibrations are transmitted away from the internal ear to prevent damage to your hearing apparatus. Okay, so like summarizing, the sound waves are transmitted from the ear pinna through the ear canal to the tympanic membrane. The tympanic membrane causes vibrations of the malleus, the incus, and the steps. The steps then sends these vibrations through the oval window to the internal ear, and the internal ear, the sound waves causes vibrations in this perilymph of the scala vestibuli. Then the vibrations are transmitted into the endolymph or the scala media. These vibrations then cause vibrations of the hair cells in the organ of Corti. The hair cells touch the tectorial membrane, and this generates nerve impulses that is carried by the cochlear nerve fibers to the brain, and thereby you have the sensation of sound, right? So this is an extremely complicated procedure through which your body detects sound. Then there is a vestibular apparatus. This vestibular apparatus is also part of the internal ear, but it does a very different thing. It is involved in maintaining body balance. So the fact that when you tip over, when you are inclined towards one side, then that is detected by this particular vestibular apparatus, which consists of 3 semicircular canals, and these 3 semicircular canals are at right angles to each other, and these are fluid filled inside, and the fluid moves inside these 3 canals, and the position of the fluid can be determined by the ears, and based on the position of the fluid inside the canals of the vestibular apparatus, your body knows the direction in which you are particularly standing. So when you are standing straight, all the fluid is at a particular level, let's say, but now let's say you tilt towards your left, then the fluid in one of the canals tilts towards the left, and the motion of this fluid is detected by particular sensors in these canals, and these sensors send the information to the brain, and your brain immediately knows the position of your body in a 3-dimensional space. And so when, let's say, you tend to slip over, so let's say you were walking and there was something slippery on the road and you were about to fall down, but immediately when you move towards one side, the fluid in the vestibular apparatus sends the signal to the brain, and the brain immediately sends out involuntary responses and tries to correct that your posture so that you do not fall or tip over. Okay, so the ears are therefore involved in 2 different functions, both for maintenance of body balance and also for hearing. Okay, so with this we come to the end of the chapter of neural coordination, and I think all different parts of the chapter are by now clear to you. So after this, I'll give a thorough reading of the NCERT book, and I'll also send you these notes, And after you've gone through all of them, I think this chapter will be very clear and you will be in a good position to answer all the different MCQs that you face in your NEET exam. Okay, thank you for now and see you in the next video.